All right, ladies and gentlemen, boys and girls. Today, I'm going to use this video to talk a little bit something called the endosymbiotic theory. All right. So, first of all, think about our prefixes and suffixes. That's a really big word, but we can break it down and get a little bit of a gist of what we're talking about here. We have the prefix bio, life, sim, meaning together, and endo, meaning inside. So we have life working together from the inside. All right. And one of the things I want to cover about in this video here is a couple things. I want to talk about what does the theory state, what is the evidence that supports a theory, because remember, a theory is an explanation supported by theory, so I'll explain what it's explaining, it's supported by evidence. We're going to talk about what that evidence is, at least the major pieces of evidence, and then finally I'm going to talk a little bit about how it connects into what we've been talking about. All right. So in our last video, we started talking about something called prokaryotes, and kind of think of like the timeline of what we believe how life started on this planet. We're kind of at this point here where the Earth is covered with prokaryotic cells. Now remember, these are very simple cells. All right. Like a lot of modern day bacteria are still prokaryotic, but at the time this is kind of all that existed on this planet. They are unicellular. They don't have a nucleus, and they don't really have organelles or extra specialized functions, structures inside their cells. And the reason for that is because, remember, they have very limited amount of DNA, fewer genes, sections of DNA. Fewer genes means less proteins, less workers means less things you can do. All right. It's not until you can get the higher levels of proteins and actually more complex proteins that you can start working together. To look back at that in our previous video talking about the central dogma of biology. But I want to talk a little bit about what the endosymbiotic theory states. All right. And basically what it is kind of what we think actually happened on this planet. So at some point on this planet, we were composed of, or the life on this planet was composed of different unicellular prokaryotes. All right, so I have a big brown one here, I have a little green one here, and I have a little purple one here. All right, and what the theory basically states is that if these were the only organisms on the planet, some of them would have been autotrophs. Maybe say this one here is an autotroph where it can create its own food using photosynthesis, but most of them were probably heterotrophs. They had to eat other organisms. And the way that an organism, or a unicellular organism, eats another organism, they don't have mouths. They're made up of one cell. And so what they have to do is basically they wrap their membrane, this outer layer, right? They have to wrap it around an object. And then once it kind of closes in on it, it pulls it in, and it has specialized enzymes, which is a type of protein, enzymes that help dissolve it. All right. The endosymbiotic theory says that maybe something different, a little different, happened in that situation here. So in this case here, let's say this larger unicellular organism was going to eat these smaller ones. All right. It would wrap itself around those organisms, right? So let's say it's just going to have a double snack here. And let me do it a little bit differently. It would wrap itself kind of around it so that it could engulf those unicellular organisms around its membrane, kind of swallowing it up. All right, and what it would do is then it would pull it in to itself. But it would still have its membrane around it. Talk about later. This is a process called phagocytosis. start to dissolve it and eat them. But now what they theorize is, those enzymes that are required, enzymes, a type of protein, to digest these smaller prokaryotes. Perhaps there were a couple organisms where the DNA was not quite right. Which means if the DNA is not right, the protein's not right. And if the protein's not right, maybe the enzymes didn't digest it. All right? Maybe it was unable to actually eat that food. You think it's like, oh, well, it's a mistake. But at the same time, now there's these living organisms inside them that are able to do extra things. And all of a sudden, this becomes a huge benefit. All right? Let's say this guy here, and I think it's in one of the video clips I sent you guys. Let's say this guy here can turn sunlight into sugar, and this guy can take a sugar and really efficiently turn it into energy. But now you have them both working for you. All of a sudden, you can create your own food and then efficiently turn it into energy. This big guy here just became better. Why? Because he had a mistake. His enzymes didn't work right. Well, if that mistake is leading him now to have a better advantage, now he can take sunlight, make his own food, and then use it, he's more likely to survive. And when he replicates and becomes two different cells, both of them are going to have that advantage. This is what we call, this is a major thing in what we call natural selection when we talk about theory of evolution, is advantages are more likely to allow you to survive, and thus you pass down that advantage. Right? Even if that advantage was originally a mistake. But sorry, I'm going away from the theory. So we have the idea that these prokaryotes were engulfed by a larger prokaryotes, and now we have this prokaryote that has smaller prokaryotes inside of it. All right, doing more complex things. Now we have much more complex organism here. All right, we have much more complex organism, and eventually this complex organism now has these complex structures inside of it, doing another job for them. These would be eventually what we would call nowadays organelles. All right, and these organelles, and now we have a much more complicated organism that has organelles or specialized structures that allow it to do more things inside of it. This eventually is going to become what we call a eukaryotic cell, which we'll talk more about in another video. A much more complex. So the idea is we have prokaryotes engulfed it to create specialized functions inside of it. All right. And these eventually we call organelles, and now we have our first much more complex cell. All right. It can do more things. It's more likely to survive. It might gain more DNA and then thus more genes and much more functions. All right. So now let's talk a little bit about the evidence real quick on this. All right. The evidence. There's three major parts of evidence that kind of support this. Doesn't have to be any specific order, but first of all, any theory is only as strong as the evidence that presents it. One thing that we see here, nowadays we would call this right here, this would be the chloroplast. The one that could turn sunlight into sugar. And the one that can efficiently turn sugar into energy, nowadays we call that organelle the mitochondria. All right. And what we notice today about the mitochondria and the chloroplast, even today, we are made up of eukaryotic cells, and we have mitochondria. Plants both have mitochondria and chloroplast. In both plant cells and animal cells, including our own, we find that they have a double membrane. Not every structure inside of our cells have a double membrane, but there's a couple of them that do. All right. And the only reason where you could suspect why that would happen is that one of the membranes is the original prokaryotic membrane, kind of the inner layer, and then the outer layer was actually that original prokaryote that absorbed it. And then when they replicate, this cell becomes two cells. They're going to be identical to each other. They keep that double layer. All right? And that the idea that some organelles specifically the mitochondria and chloroplast, have a double membrane. That double membrane, one from the original prokaryote, and then one from the other prokaryote when it engulfed around it. All right. Number 2. So we see that in a lot of organelles. That tells us something about it. The second thing that we notice is that that when cells reproduce, all right, when a single cell reproduces, even a single cell complex eukaryotic cell reproduces, it makes an identical copy of itself. All right, in the fact that it replicates itself, but the way that the mitochondria and chloroplasts, they replicate not like organelles, but actually more like prokaryotic cells. All right, and so that the way they do it, they basically reproduce. Sorry, just trying to think how I want to say this. The chloroplast and mitochondria, which now we know are these organelles, reproduce like prokaryotes, which we'll talk about more about how that happens when we talk about mitosis. All right. And the third piece of evidence is a big deal. All right. And each one of these are prokaryotic cells. They have DNA. Right? They would have some DNA. That one has DNA. This one has DNA. All right. And even in today, in eukaryotic cells, we actually find, the eukaryotic cell now has a nucleus that contains its DNA. But we actually find separate DNA in our mitochondria and in our chloroplasts. We don't really use that DNA. This is the DNA we mostly focus on now, the one that's in the nucleus. But we find mitochondrial DNA. And it's very, very specific. Mitochondrial DNA. We have DNA in our mitochondria. It's not its function. Its function is not to store our genetic information. That's the job of the nucleus. But yet it still has it. Why would it still have DNA, if it's not its function? And it's because we think it used to be a prokaryote, and it's just kind of still there, even though it's not doing that job anymore. All right? I used to work at a factory for a while when I was in college. I still have the uniform I have to wear. Even though I don't use it, I still have it, and it tells me something about the past. And that's what we have here, where you can tell something about the mitochondria's past because something's in there. So both the chloroplast, I'm going to abbreviate, and mitochondria both have their own DNA. Separate from the rest of the cell. All right, so now how does this all tie together? Why does this matter? All right, because we're still kind of in that building block of how do we go from non-living molecules into complex multicellular organisms. And we've talked a little bit about the introduction of DNA, creating proteins. The more genes you have, the more proteins, the more complex you could be. You could go from single-cell prokaryotes into multicellular eukaryotic cells. We've gone from very simple forms of life, and now we've created, still in this picture here, still unicellular right now. But now we've created a cell that has a lot more going on inside of it. Are a lot more complex, which means if it's a lot more complex, it can do a lot more things, including to the point where it could become multicellular, all right, and do much, much more complex or functions. So this is the theory of this is the endosymbiotic theory, or the theory of endosymbiosis, the idea of how prokaryotes could have absorbed other prokaryotes to create a much more complex, eventually leading to modern-day eukaryotic cells. Okay. And of course, our evidence to support that. All right, that's going to be it for this video here. Go back through it. Make sure I put those questions to kind of think about ahead of time. Can you answer those questions? If not, go back and kind of try finding the video where we left off, and I talked a little bit more about that. Otherwise, thanks for watching. Good luck, and may the science be with you. Go Browns.